Assalamu alaikum students. I hope that you are fine. Let we discuss the topic is Mucor. Mucor is belong to a kingdom fungi. Let's use the diagram of Mucor. Taxonomic position of Mucor. Kingdom, division, class, order, family, genus. Kingdom fungi. Division Mycota. Class Zygomycota. Order Mucorales. Family Mucorrhiza. Genus Mucor. What you see habitat of mucor. It is appears rarely in damp horse dung when kept under a bell jar. Present soil, bread and other food materials. What you see there is diagram. Vegetative structure of mucors. Mycelium consists of numerous slender, freely branched filaments called hypha. The hypha form a fully mass in color called a mycelium. After mycelium formation, reproductive phase starts. Black pin-like structures appear on mycelium card as sporangia. The hypha are coencytic and aseptate. At vegetative stage, the hypha consists of slow horizontal hypha and rhizoidal hypha. The rhizoidal hypha has seen the absorption of nutrients from the substrate. Cell wall is made up of chitin and cellulose is absent. The organelles like mitochondria, endoplasm, reticulum are present in cell. You see the structure of mucus, sporangia, rhizoids, sporangium, sporangiospores, rhizoids, stem. You see this diagram. Mitochondria, ribosome, vacuole cell wall, nucleus. That you see. This septum sporangiosphore substrate mycelium, apophysis, cell wall of sporangium, sporangiospores. You see this diagram. Vegetative reproduction of mucor fragmentation. When the separation of hypha occurs from the oleo portion, then these fragments are capable of producing new hypha, and this is called fragmentation. Chlamydospore formation. Under unfavorable conditions, the fungus produce special modified thick word dressing cells called as chlamydospores. They contain food and are very resistant to desiccation. When addition conditions become favorable, it germinates to form new mycelium. What you see diagram of mycelium. First A, B and D. Asexual reproduction, sporangia, sporangiospore. It takes place the formation of non-motile multinucleate sporangiospore. These sporangia arise singly and terminally at the tips of sporangiospore. The young sporangium is devoid physical structure. Admixturally, it roots into black color due to formation of spores. In the center of spore, there is large dome-shaped structure called coelomia. The sporangia are haploid. They are produced in hypha, mycelium, and produce haploid spores. Asexual reproduction. Let's use the diagram. First of all, sporangium. Second, sporangiospore. Third, coelomia. Aerial hypha, sporangiospore. Posterior hypha, subterminal hypha. Structure of mucor. What you see structure of mucors? You see spores. Spores are dispersed after the reporting of sporangia. They occur dispersed by one. The spore is non-motile, oval, and contains protoplasm surrounding by single layer cell. Under suitable conditions, the spore germinate to form a germ tube. The hypha arrives from the germination tube and spread in all directions. Diagram of Mucor. Let's use this is round up and small. Mucor. Sexual reproduction. Ictway place. Germination copulation. The few in germination are multicellular and are distinct for each other. The sexual process starts with the formation of two special hyphae called zygospores. They arise from near the apices of activity growing somatic hyphae and negative and positive strands. They are commonly induced. The zygospore meet and adhere in pairs at the tip to form fusion septum. The stem cells due to flow of nucleic acid cytoplasm in them called as prognathongia. Prognathongia combine to form a prozygosporangium containing many diploid nucleic acids. Zygospores. Let us see diagrams. Prognathongia, negative and positive hyphae. Zygospores, gametongia, prognathongia. Let you see this structure and you see complete structure of mucosexual sexual cycle. First, a vegetative mycelium, protestor hypha, germination positive and negative divided to fragmentia positive and negative is divided into young synsporium, germination psyllium, Spencer, zygospore Spencers, germinating zygomes, germination sporangium, germination spores, meiosis, and at last meiosis germination and is divided A and B, and vegetative mycelium and posterior hypha. This is complete structure of mucors. Students, this video is end. Okay, Khuda Hafiz.